Hi. I'll briefly summarize in this video a recent work done in collaboration with Willem van der Linde, Enrico Scorupa, Pauline Kolbeck and Jan Lipfert. The paper can be downloaded from bioRxiv. In magnetic tweezers experiments, the two ends of a DNA molecule are attached to a solid surface at one side and to a small magnetic bead at the other side. External magnets are used to apply a stretching force. By rotating the magnets, one can change the DNA linking number in a controlled way. If the number of turns exceeds a given threshold, the molecule buckles, forming an intertwined loop structure known as plectoneme and shown in the left figure. In magnetic tweezers experiments, one measures Z, the extension of the molecule, which is the distance bead surface, as a function of time and various parameters. The middle figure here shows some traces of extension versus time of bare DNA at a fixed force and three different linking numbers. When delta Lk is increased, the average extension drops, a well-known effect. The topic of our paper is the analysis of the variance of the extension, which measures the fluctuations. While the average Z decreases when increasing or decreasing delta Lk from the torsionally relaxed state, delta Lk equal to 0, the variance of Z increases, see right figure. To understand this behavior, one can use the two-phase model developed by John Marco and collaborators. Instead of delta Lk, it is convenient to use here the supercoiling density sigma, which is an intensive quantity proportional to delta Lk. The inputs of the two-phase model are the free energies of the stretched and of the plectonemic phases, shown here as dashed line as function of sigma. At small sigma, the free energy of the stretched phase is smaller than that of the plectonemic phase. At large sigma, the opposite happens. At small sigma, the free energy minimum corresponds to a pure stretched phase. This is the pre-buckling regime. When sigma exceeds a threshold value, sigma s, plectonemes appear. The minimal total free energy is obtained from a double tangent construction. This is the post-buckling regime of phase coexistence between stretched and plectonemic phases. The coexistent region ends at sigma p. Beyond that point, the molecule is fully plectonemic and the end-end distance vanishes. The expression for average Z shown here is derived from the two-phase model. It is characterized by two parameters, gamma, the slope, and sigma p. The figure at the bottom of the page represents the two-phase model with the stretch phase shown in red and the plectonemic phase in blue. The two phases are characterized by average supercoiling density, sigma s and sigma p respectively. Summarizing, the two-phase model predicts that the free energy is, in the coexistence region, linear in the supercoiling density sigma, equation 1. As a consequence, the average of Z and the variance of Z are also linear in sigma, equations 2 and 3, because they are derivatives of the free energy with respect to the force. As we have seen, the average extension is a function of two parameters, gamma and sigma p, equation 2, which are both force dependent. Differentiation of the average Z in F give rise to equation 3 for the variance. More insights on the origin of fluctuations can be inferred from Monte-Carlo simulations. We show here simulation data of the twistable worm-like chain model of DNA at fixed force and supercoiled density sigma or delta Lk. The figure C shows an histogram of values of Z from equilibrium sampling. The figures A and B are snapshots of extreme conformations with very small and very large Z respectively. Stretched and plectonemic phases exchange lengths, which leads to the fluctuations in Z. It is also possible to understand why the fluctuations increase when increasing sigma. We don't discuss this issue now. We come now to the core of the paper. Why are we interested in fluctuations? We will show how the variance of Z provides quantitative information on DNA-protein interactions which are not accessible from the average of Z. Consider a protein that bridges across two sides of a DNA molecule. This is likely to bind in the plectonemic phase, where two sides come in close vicinity. Here we do not show a protein, but two linked coarse-grained beads in red in our simulation model. Once bound, the protein splits the molecule in two domains, a looped domain of length delta L, dark blue in figure, and an unlooped domain of length L minus delta L. We now use the two-phase model to describe the unlooped domain and derive the changes in average Z and variance of Z upon protein binding. How does the average Z change upon protein binding? To calculate the average Z for the unlooped domain, we use the two-phase model expression, replacing L and sigma with new values L star and sigma star, the length and supercoil density of the unlooped domain. A simple calculation shows that the product L times sigma p minus sigma does not change. In conclusion, the average Z does not change upon protein binding. Hence, delta average Z is equal to 0. The variance has also a term proportional to L times sigma p minus sigma, which does not change upon protein binding. However, the term gamma times d sigma p df times L does change. The effect is that the variance drops upon protein binding. This drop is proportional to the looped domain length delta L. The result is rather intuitive. The protein prevents the plectoneme from opening beyond the binding point. The exchange of lengths between plectonemic and straight phases is reduced, which leads to a decrease in the variance. This idea was tested in Monte-Carlo simulations. Figures A and B in the left show the average and variance as a function of delta Lk, or sigma, see top horizontal axis. Now we select one value of sigma in the post-buckling regime. This is sigma equal to 0.04, red circled in figure A and B. We perform simulations at that fixed sigma. We randomly select two beads in close vicinity in the plectoneme, in red, and generate a loop domain of given length delta L. This mimics a protein binding event. We continue the simulation with these two sides frozen and sample average and variance of Z. We repeat this for many different events, generating loops of different lengths. The results are collected in figure D and E. We see that the average Z does not change, but the variance decreases linearly with the looped domain length, as predicted by the thermodynamic model. The dashed line in figure E is not a fit, but it has a slope that can be estimated using linear extrapolations of the data of figure A and B. The details are in the paper. Experiments were performed using 3 enzymes with specific binding sequences shown in the table. They are chosen such that there are only 2 binding sites on the DNA construct. The positions of these sites along the DNA using magnetic tweezers experiments is known, therefore we know delta L, the looped domain length. The first enzyme, BSAX1, has 2 binding sites which are close to each other, producing a small loop of 321 base pairs, or 123 nanometers. SAC2 produces a loop of length 2382 base pairs. We show here time traces of Z and its standard deviation for the 3 enzymes. We plot standard deviation and not the variance for convenience, because the standard deviation has the same units of Z, micrometers. Enzymes are added to the solution at time t equal to 0. The enzymes have no effect on average Z, but there is a drop in the standard deviations, or variance. The effect is small for BSAX1 and large for SAC2. The next page shows the same figure of trace versus time. The right figures collect the data for average Z and variance of Z as functions of the expected loop length. Experiments are replicated about 15 times for each enzyme. The data follows quite closely the thermodynamic theory. There are 3 out of 13 points and these are outliers for SAC2, indicating a possible alternative binding mode. We said that the protein binding does not affect the average Z. However, after protein binding, we observe in particular in the enzyme NAE1 some transitions in the traces Z versus time. These appear as discrete steps. The interpretation is that these are partial dissociations of the protein and reassociation. Reassociation can lead to a linking number exchange between looped and unlooped domains. We can test this hypothesis using Monte-Carlo simulations. The experiment shows 1 most populated state and 4 less populated states corresponding to a linking number exchange of ±1 and ±2. Linking number exchange in the protein can only involve change by integer values. However, in Monte-Carlo, we can also simulate reassociations with a continuum linking number exchange. There is an excellent agreement between Monte-Carlo data and experiments. Monte-Carlo model has no adjustable parameters here. Finally, from the variance distribution of linking number exchange, we can estimate the torsional stiffness of the plectonymic phase. We find P equal to 20 nanometers, consistent with previous measurements using different techniques. In conclusion, we have strong indications to believe that the steps in Z observed in the experiments are really dissociation-reassociations events with linking number exchange. This concludes a summary of the main results of our paper. If you have any questions, comments related to this work, please contact us.